Good morning. Today we'll discuss the program BSc Botany, Semester 3, and the session is 25. The anomalous secondary growth in Dracaena. The growth in plants occurs as the stems and root lengthens. Some plants, especially those that are woody, also increase in a thickness during their lifespan. The increase in the length of the shoot and the root is referred as primary growth. The primary growth it is a resultant of cell division in the shoot apical meristem. The secondary growth is characterized by the increase in the thickness or the girth of the plant. It is caused by the cell division in the lateral meristem. The herbaceous plants mostly undergo the primary growth with a little secondary growth or increase in a thickness, whereas the secondary growth or wood it noticeable in a woody plant, it occurs in dicots, very rarely in case of monocots. The primary growth in woody plants, the primary growth is followed by the secondary growth, which allows the plant stem to increase in a thickness of the girth. The secondary growth or the secondary vascular tissues is added as the plant grows, as well as the cork layer. The bark of a tree extends from the vascular cambium to the epidermis. Some plant parts such as stems and roots continues to grow throughout the plant's life, a phenomenon which is called as the indeterminate growth. Other plant parts such as leaves and flowers exhibit determinate growth, which ceases when a plant part reaches that particular size and shape. Most primary growth occurs at the apex or tips of stem and root. The primary growth is a result of rapidly dividing cells in the apical meristems at the shoot tip and root tip. Subsequently, the cell elongation also contributes to the primary growth. The growth of the shoot and root during the primary growth enables the plant's body to continuously seek water through the root, sunlight through the shoot. The influence of the apical bud on overall plant growth is known as the apical dominance, which diminishes the growth of the axillary bud that forms along the sides of the branches of the stem. Most coniferous trees that exhibit the strong apical dominance, thus producing the typical conical shape, that is what you call it as Christmas tree shape. If the apical bud is removed, then the axillary bud will start forming the lateral branches. So the gardeners make use of this fact when they prune plants by cutting off the tops of the branches, thus encouraging the axillary bud to grow out and giving out the plant like a bushy shape. Coming to the secondary growth, secondary growth, it is an increase in a thickness or girth by cell division in the lateral meristem, what I explained or what I mentioned in the first slide, called the vascular cambium and the cork cambium. Not all the plants exhibit secondary growth. The secondary growth is formed by the formation of the vascular cambium and the cork cambium. Vascular cambium is formed by the joining of intra and intravascular cambium. The cells divide periclinally to produce secondary vascular tissues and ray parenchyma. The secondary growth increases the growth of the plant of a root and stem rather than its length. In many of the plants, the pattern of secondary thickening showing the deviation from the normal type. That's why the term anomalous secondary growth is given for this deviation. The anomalous secondary growth is common in tropical plants in case of dicotyledons. The secondary growth or woody, it is noticeable in woody plants. The secondary growth is usually present in case of monocots and absent in case of the secondary growth is usually present in dicots and absent in monocots since the vascular bundles are of closed type. A few plants in case of monocots which exhibits the vascular bundles closed type. Along with this, they shows the anomalous secondary growth, example Dracaena and Ecca. The reason for the secondary growth is normal cambium behave peculiarly or irregularly, resulting in the abnormal arrangement of vascular tissues. Normal cambium is situated in an abnormal position, hence the tissues cut. If it is present, it is replaced by the additional accessory cambium rings. So, we'll come to the actual topic, that is anomalous secondary growth in Dracaena. Anomalous secondary growth in Dracaena, it is a typical example of anomalous secondary growth in monocots. Typically, secondary thickenings is absent in case of monocots. Therefore, the secondary thickenings itself is anomaly as Dracaena is a monocot. The Dracaena exhibits the primary structure, that is epidermis. The epidermis is an outermost consisting of a single layer of regular parenchymatous cells. The hypodermis is situated below the epidermis, which is composed of sclerenchymatous cells. The ground tissue is undifferentiated parenchymatous, numerous, closed, which exhibits the secondary collateral vascular bundle scattered in the parenchymatous ground tissues, and the vascular bundles are closed type scattered in the ground tissues. The xylem is endark. Coming to the anomalous secondary growth thickenings in Dracaena. In Dracaena, the cambium appears in the parenchyma outside the outermost vascular bundles, the region in which the cambium appears in sometimes as cortex and sometimes as a pericycle. In Dracaena, the secondary growth is due to the extrastellar cambium ring in monocot, stem at the cortex, and abnormal activity of the cambium. The epidermis, it is a single layer barrel-shaped structure with a cuticle followed by the phellum and the cork cambium. The phellum is, otherwise it is known as cork. Without intercellular spaces, it has been arranged, and the cork cambium. The secondary thickenings is initiated by the formation of a special meristematic zone in the inner cortical region of the stem. A multi-layered cambial ring is differentiated from the inner cortical cells laying outside the vascular bundles. This cambium is formed in the region of the stem which sees its elongation. The shape of this cambial cells varies. The activity of this cambium is abnormal. In a cut, it cut a large number of cells to the inner side and a few amounts of cells to the outer cells. The cells produced external to the cambium are parenchymatous. The cells produced inner to the cambium are partially parenchymatous and partially vascular in nature. The parenchymatous cells produce internal to the cambium, which is developed into lignified conjunctive tissues. The vascular tissues produced internally to the cambium, which is differentiated secondary vascular bundles. The vascular bundles are concentric amphivasal type. The cells of the inner cambial ring participate in the formation of the secondary vascular tissues. So, the cambial ring is formed due to the meristematic activity of some cells lying immediately outside the bundles. Immediately outside the bundles, the cambium cells are unusual in functions, which go on producing secondary vascular tissues and conjunctive parenchyma internally and in little simple parenchymatous cells externally. The secondary vascular bundles formed are oval and amphivasals and are smaller than the primary bundles. They are embedded in the conjunctive tissues whose cells are radially arranged. In the extrastellar regions is formed because of the repeated periclinal division of the cortex cells. The cells are formed without the appearance of the cork cambium called storied cork. And later division, they are irregular to form of mass of cells. In this mass of cells, the centrally placed cell metamorphosis into phloem cells, the peripheral cells differentiated into xylem elements. Thus, the newly formed secondary vascular bundle consisting of centrally placed phloem, which is surrounded by the xylem, concentric vascular bundle, that is amphivasal type. The cambium then cut parenchyma cells internally. This process continues and many rings of concentrically arranged vascular bundles are formed. The last ring of vascular bundle is embedded in the mass of lignified conjuncting tissues. A cork cambium may also develop, and this cork cambium can produce the bark. So, the overall conclusion of this session is the secondary growth is which is exhibits usually in dicots and very rarely in monocots. That is the outward growth of the plant, making it thicker and wider. The secondary growth, it is very important in the woody plants because they grow much taller than other plants and need more support in their stems and root. So, lateral meristems are the dividing cells in the secondary growth and hence produces secondary tissues. Thank you for listening.